Welcome into the third part of this video, which is applications of biotechnology. So in this lecture we are going to study how to make a genetically engineered insulin which is helpful for the patients who are suffering from diabetes. So if you want to know all this information, watch this video completely and subscribe to my channel will be incredible. Genetically engineered insulin. Due to the deficiency of insulin, diabetes mellitus occurs to the people. For the treatment of this disease, in earlier time, insulin was extracted from pancreas of cattle and pigs. This is because in earlier time biotechnology was not developed well, so in earlier time to get insulin, people extract insulin from pancreas of cattle and pig, and that insulin was generally unsatisfactory and allergic to the some peoples. To treat this problem, a large amount of insulin is required, and insulin which is like human insulin was also required. Now let's understand how genetically engineered insulin is formed. For this we have to study about the structure of insulin. So here in the right side of the video you can see the structure of the insulin. Here you can see the first structure which is formed is proinsulin, where three chains are here: chain A, chain B and chain C. This is proinsulin, and now its maturation occur where only chain A and chain B are present, which are connected by disulfide bond, and chain C is free. So this chain A and B which is connected by disulfide bond is a mature insulin, and this is a main hormone which is used for the treatment of diabetes and which regulate the sugar level in the humans. So in 1983, Eli Lilly, an American company, made this mature insulin in external medium. To form this mature insulin, they prepared two DNA sequences. These two DNA sequences code for A and B chain of the insulin, and these DNA sequences then inserted into the plasmid, which is PBR322. We have already studied in our previous video that in PBR322 only one selectable marker is present, which is lag-ZAD gene. So this is inserted, these two DNAs are inserted into the lag-ZAD gene, so we can separate them from blue color solutions. And now these plasmids are inserted into the E. coli. Now E. coli produce separate A and B chains, and these two A and B chains of insulin are connected by disulfide bond. So this mature insulin is formed by this complete process where two DNA sequences are made for the chain A and B, which are inserted into the plasmid. These are inserted into E. coli, and E. coli produce separate A and B rings, and these A and B rings are connected by disulfide bond. So this is the complete procedure of making of mature insulin, which is like human insulin and which is help for the treatment of diabetes mellitus. Now an important point is comes with respect to insulin that can be asked in exams very too. That is very important, that is insulin cannot be taken orally. That means we cannot eat the insulin tablets or anything else, because these are digested in our digestive system. To get insulin inside our body, we have to inject these insulins in our muscles, so that a little amount of insulin is secreted every time in our body, so that our sugar level is maintained. If we directly inject too much insulin in our body, so this will decrease the sugar level in our body, and which is dangerous for us. So this is the properties of insulin. So if you watched this video, like this video and subscribe to my channel will be incredible. Thanks for watching. See you in the next video.